I'm Patrick from RedShiftBio Analytics, and today I'm going to give you a presentation on an application note that's available on our website entitled Biosimilar Structural Comparison of Commercially Sourced Reference Standards by MMS Rapidly Detects Subtle but Critical Differences to Correctly Predict Activity for Use in an ELISA Product. So if we look at proteins, these are complex three-dimensional structures very often for which their structure and function are directly related, in some cases critically. For example, here shown is bacteriorhodopsin on the right-hand side, or perhaps for actin, shown in the bottom left, or perhaps for a calcium-binding protein, calmodulin, shown on the right-hand side here. For each of these, a structural change either at the binding site itself or very often at a different part of the molecule entirely can affect the binding of its preferred ligand to the protein target. In this case, we studied bovine serum albumin, or BSA, because this is used as a control molecule in an ELISA test kit assay. We compared a known functional reference sample of this BSA to 13 other samples from different manufacturers and different batches from those same manufacturers, some of which were shown to function correctly in the assay and some of which were shown to function incorrectly. To do this, we used a novel form of infrared spectroscopy called microfluidic modulation spectroscopy, brought to you now in the Apollo platform launched in May 2022 by RedShiftBio Analytics, which is an automated platform that allows you to collect extremely sensitive infrared-based structural data for your proteins from either 24 or 96 well plates with very little user interaction and to a much higher degree of sensitivity compared to traditionally available technologies. So when we make our measurements with MMS, very much like the Beer-Lambert law in UV spectroscopy, as we increase the protein concentration and make absorption measurements within this amide 1 band region, we see an increase in the signal or the absorbance of the sample as we increase the protein concentration, and this relationship is linear. If I plot a serial dilution of one of the BSA samples shown, the reference sample in fact, we see that this linear relationship is extremely robust, and really this highlights the reproducibility and sensitivity of the infrared measurements that are possible using this novel technology, especially if we look at the R-squared value of the fit to this line. In this case, for this experiment, this was 0.99992, which I hope you would agree is extremely high. With MMS, we were able to make measurements, quantitation measurements from 0.01 or structural measurements from 0.1 milligrams per mil, all the way in both cases to 200 or above milligrams per mil in a very wide range of different backgrounds as well. And we retain this extremely high sensitivity and reproducibility of the measurement, which is very key particularly when you're trying to determine very, very small differences in structure between a range of samples, such as for this study. So the first thing we did when we took our 14 BSA samples, our 13 samples from different manufacturers or different batches from those same manufacturers, compared to the reference standard that is highlighted in yellow here, we were told that each of these samples were 10 milligrams per mil in concentration. So first of all, we compared the spectra we generated to a reference of known concentration to more precisely calculate the concentration of each sample. We then did a similarity analysis of the overall shape of these different spectra, particularly looking here at the second derivative spectra in each case. And this allowed us to very clearly distinguish these 14 samples into two groups. One group shown in dark blue, which is most structurally similar to the reference, and a second group that is less similarly structured to the reference, shown in light blue. Although I should note the difference in the overall structure of these two groups is only 1% in this case, approximately, which is an extremely small difference, one that would be very, very challenging to measure using traditional technologies. If we deconvolute the second derivative data by doing some curve fitting analysis, we can deconvolute this data into a much simpler output. In this case, a bar chart output showing you the breakdown of secondary structures across turn, alpha helix, disordered, and beta structure. We can see that the reference in purple shares a very similar structure as we saw from the similarity results to all of the other samples, and it's not immediately clear that there's a pattern that would separate the reference sample that is known to function correctly from those of the other samples that did not function correctly. If we zoom in on all of these, we see that this is true for alpha helix, for beta sheet, and for turn structure. But actually, if we look very closely at disordered structure, we do see that there is a pattern here. In purple, near the right-hand side is the reference standard, and you can see that 5 of these samples do actually show a similar level of disordered structure to this reference, and the other samples show a small increase in disordered structure of about 1.5%. Actually, another way we can look at the data, and in this particular case, I think the best way to look at this data is to interrogate the raw data itself. So what I've done here is I've taken the second derivative spectrum of our reference sample, and I've subtracted each of the second derivative spectra of the other 13 samples from it to create a difference plot that we term a delta plot. So in this case, any samples that follow closely to the baseline should be structurally similar to the reference, and any that deviate from this baseline should be structurally different to the reference. And indeed, we do see this separation. We see the same 5 samples we saw in the pattern for disordered structure following somewhat closely to the baseline. There is some variation, which is why I think we see some variation in the other secondary structure outputs, and very clearly a second group which shows an increase, particularly in this disordered structure, but more importantly, is structurally different to the reference that is known to function correctly. We can actually, if we take the absorbances at each of the peak positions here, we can actually display this data in a line plot output, which I think would be most suited as a QC output if you wanted to look through a very large number, particularly of samples, and compare their structures very quickly to a reference like we have done here. We can see very clearly here that we have 2 groups. The ones, if you look particularly at the orange line, that follow the baseline very closely, the same group I showed you before, and the second group that shows a structural deviation from the reference that was known to function. And indeed, the group that shows the highest structural similarity to the reference, the ones highlighted down the bottom here, were indeed shown to be functional in this ELISA assay, whereas the second group that shows this approximately 1% change or difference in structure to this known functional reference were shown to be inactive or incorrectly active in this ELISA assay. So thank you very much. If you would like to read about this application note in more detail, you can find a link on the right-hand side of the screen. And please do visit our website for a much wider range of application notes, publications, and other videos to teach you more about the MMS technology. Thank you.